And there'll be a longer break for lunch. We're not going to make you like eat really fast. So, okay, and so the next speaker is going to be introduced by Alex Song.
Hi everyone, my name is Alex Song. I'm a PhD student in Raskin Lab in Environmental Engineering. And today it's my honor to introduce Professor Ellen Decaestecker. She earned her PhD from University Basel in Switzerland, and now she is a professor in Department of Biology from KU Leuven, Belgium. She also serves as the chair of Interdisciplinary Research Faculty at the KU Leuven and the secretary of European Society of Evolutionary Biology. Her research group explores host microbiome interactions across invertebrates to primates, examine how environmental changes drive disease and evolutionary dynamics. Using molecular tools, they investigate how microbiomes and epigenetics shape resilience to stressors and genetic signals in the human microbiome to reveal evolutionary links. Their work connects ecosystem health with adaptability across species. Today, she's going to use Daphnia and its parasite as a model to investigate the evolutionary ecology of host-parasite interactions. Let's welcome Professor Ellen Decaestecker. Thank you. Sorry, the microphone is rolling.
Okay. So thank you very much. Thank you very much for having me here and for the invitation. It's really a great experience to be back in the United States. It has been a while. So indeed, on the slide, you see the Daphnia model. We're working on more models. And actually, we'll start my introduction more from a general perspective, so on the role of the microbiome in eco-evodynamics. So as you see here, we actually put microbiome very central, and the microbiome, how it interacts with hosts and modifies host phenotypes, so structures eco-evodynamics. So, and I will use a bird to show something else than Daphnia to showcase that. So as we all know, organisms have microbiota in the gut, also at other places. But let's focus a little bit on the gut now, and there we have a very interactive microbial community, and we have processes there already, eco-evo interactions, so ecological and evolutionary interactions, and very crucial in eco-evolutionary interactions is actually that they are occurring at the same time and actually also influencing each other. So it's really feedback loops between the two different systems. So we know microbial communities can be modified by host factors. Also very importantly, they can be modified by environmental factors. I will tell a little bit more about that. And of course, you also have very strong microbiota interactions. So that's all happening there in this little space of the gut. But important to know is that even if you have this little space in the gut, it also affects your phenotype. And there the host is really crucial. So how the microbiome is modifying the host phenotype. So let's put that a little bit smaller and then see how that may modify your larger system, because of course the host is also part of a bigger community and all biotic interactions around it. So it's very crucial to understand that this microbiomes modifies host phenotypes and that the host is part of that community, and also at that level you have ecological and evolutionary interactions. So they interact ecologically, you have species distribution changes, evolutionary processes. So, but what you actually said is that you have this bottom-up feedback. So the microbiome will modify your host from a bottom-up first part of the feedback, but also secondly, you get interactions from your wider environment to your microbial community. So you have continuously loops of bottom-up feedbacks with top-down feedbacks. And that's actually one of the ideas Broos and me, and also together with Luc de Meester and other colleagues at KU Leuven, we wrote down, and actually to say like, this microbial modification of your host is really a crucial factor in that. It's often overlooked. We focus on host genotypes, but we often forget that this microbial community is a big part of that and can actually structure all these dynamics. And here you see the different levels where eco-evo interactions typically occur. Okay, so that was the bird. So moving back to the aquatic ecosystems now. So one of the topics that I said, we know that environments are changing. We are in changing climates, and that will impact your host. So we know aquatic organisms are suffering from a lot of stressors. There's toxicity present. There are parasites present. There are predators present. And that can all affect, first of all, your host, but then secondly, also microbial communities. And that's actually what we're investigating on a smaller scale in this Daphnia model. So there are actually two crucial factors I want to give you in the first part of the talk, then I will show you how we extend that. But first of all, in this whole eco-evo dynamics, what is the role of the microbiome? We hypothesize it's a missing link in this eco-evo dynamics. And then secondly, how is this missing link actually realized? It's via this genotype microbiome environmental interaction. So it's very important to see that actually this host genotype microbiome interaction, it's that modifies your phenotype. So you have modified phenotypes, and Daphnia is a very nice model for that. You know that maybe already, but they have this typically helmet formation that they do to escape predators. They are very phenotypically plastic, so you can modify them relatively easy. And then one of the hypotheses we have is that the microbiome is a big part in this phenotypic plasticity modification, and in a way that it enables the host to interact with its environment. And then the different environmental stressors we are looking at is, first of all, the cyanobacteria, especially toxic cyanobacteria. So it's part of the Daphnia food. They filter it, they get it in the gut, and we look how does the microbiome react to that, and can we use the microbiome actually to increase tolerance towards these stressors. We also look to parasites. I looked to use that before, and starting to do that a little bit on the microbiome again, and then also hypoxia and microplastics is also there in the environment. So you have a lot of different factors influencing them. But okay, to be able to do that, you have to have a model, first of all, that is phenotypically plastic, so that is fine, we know that. But then secondly, and that's a very good trait of Daphnia, is that we can keep the genotype fixed. So you can have clonal lines, so we have fixed genotypes. That's also a very important point. But then also thirdly, we have to be able to work with organisms where we modify the microbial community, or able to modify the microbial community if we want to do inoculation experiments and to see what effect your adapted inocula have on your organism. And that's why we started to develop. So in here you see the Daphnia. We take the parthenogenetic, the asexual eggs out of the Daphnia. We sterilize them at a particular time window in the development. So, and then we hatch them. And then what you can do is take the gut of another Daphnia. Mostly that's then a Daphnia that we assume that is adapted towards a certain condition. And then we integrate it again into the axenically made individuals. And in this way, we are actually able to do this transplant experiment. So we give new bacterial inocula to the axenic individuals and then investigate upon particular stressors, does it modify the phenotype, yes or no. So we do this experimental transplants in combination with sequencing, so more amplicon sequencing, and then now also recently we start to more functional profiling to be able to know what exactly that microbial community is doing upon these different stressors. I'm going a little bit too fast. So crucial questions here is if we have our Daphnia gut, so the bacterial pool we find, where does it come from? Is it the environment that determines what microbial pool you will have in your gut? Does it interact with the host genetics? Our hypothesis is yes, so you will see, we will find some evidence for that. And actually that's what we do in these exposure experiments, investigating host genotype effects versus microbial inoculum environmental dependent effects, and what do we find back in the Daphnia. So as I said, we do that via different experiments, and ideally we can work with different clones. So what you see here are 1, 2, 3, 4, 5 different Daphnia clones, so we can keep the clonal lines fixed in the lab. And then the first thing we did with Martin is actually exposing the Daphnia lines toward the Daphnia genotypes towards different microbial inocula. So we took the microbial inocula from 3 natural systems, and then one was a Daphnia inoculum. And the first thing we did is we inoculate that in sterilized Daphnia and see what is the main factor determining your community. So what is the relative impact of the bacterioplankton? So that's the community that is around the Daphnia, so the inoculum, and is there also an effect of the host genotype? So does it matter in which host's genotype the microbial community is going? And what you see here are actually the results of the sequencing of the guts of the Daphnia that got the microbial inoculum. So from the 4 different environments. So if you take the gut out of the Daphnia, you sequence it, and then you determine the microbial community. What you see is clearly, so especially this hay extract, which was the microbial community from the lab culture, clearly differed from all different other communities. You can also see that here. It's a particular strain, Citrobacter, that apparently seemed to be quite beneficial for Daphnia, that strongly determined that community shift. So definitely it matters what microbial community the Daphnia gets. So what you give, they will also take it up, at least partly. So that's important to know if you want to modify your phenotype, that it takes up the strains that you give. So that's the first thing. So the answer there is yes, but then very interestingly what you see here, so in the bottom are the 5 different genotypes, and in here there were like the more dominant bacterial strains we found back in those communities. So the bigger the dot is, the more abundant it is. So there clearly you see that the different genotypes also picked different strains. So there's also a strong host genetic background. So different genotypes pick up different strains. So actually our second assumption is also fulfilled. So we see that inoculum has a strong effect, host genotype has a strong effect. And then actually what we also saw, and that's also quite interestingly, is that so the diversity, the microbial diversity that was present in your inoculum is also translated in what you find in the microbiome. So if you give them a more diverse inoculum, they will also get a more diverse microbiome, Daphnia microbiome. And then we know from another experiment that that's also translated in performance parameters. So we see that the more diverse the microbial inoculum is, the larger the Daphnia were. So you see that having a more diverse microbiome for Daphnia is a good thing in terms of growth rate. So they are getting larger. So that's already first indication we had that microbial communities could also be translated in performance traits, which is important if you look to this phenotypic interaction. Okay, so that's a little bit the first part. So what did we see? We see, well, we have Daphnia, we inoculate them with bacteria. We see that in the microbiome, there's a strong effect of your environment. There's also a strong effect of your genotype. So we see that both factors structure our microbial community, microbiome community. But then actually for this eco-evodynamics to occur, remember that I started with this bottom-up hierarchical feedback mechanisms. We should actually also see that that whole phenotypic modification is translated in environmental changes. So the question is that if we find phenotypic modified Daphnia due to the microbiome, do we also see that reflected in our environment? Otherwise said, can we use Daphnia as an ecosystem engineer to modify your environment? And important here to know is, I didn't tell that yet, but Daphnia is really a key player in your ecosystem. So it filters everything that it passes, inclusive toxicants, but it's very important to keep your system in a healthy state. So you need Daphnia, or you need zooplankton in general in aquatic ecosystems, and you need healthy ones. So they should be able to do the filtration effects. Wait, that's wrong way. Good. So, and that brings me a bit to the second part. So now already we know, okay, genotypes and inocula determine Daphnia microbiomes. The question is, does that help for phenotypes? We more or less already, yes, we see performance effects. And then secondly, what is the role in stressor interactions? And one particular stressors we did a lot of research with is, and it's also important for climate change, is this toxic cyanobacteria. So a lot of you in the audience know already what toxic cyanobacteria are, but we see that with this more increased temperatures, more nutrients, especially more runoff of fields, especially nitrogen, we get more of this toxic cyanobacteria. And for Daphnia, that's part of their food. So that's another bacteria they just filter. So it's there in their microbial community. So it's actually one of the competitors in the microbial community. So we started to wonder, okay, can Daphnia interact with this cyanobacteria? It's a predator-prey interaction. And do we see changes there? And does the microbiome play a role? So one of the things, so Maxime, Emma, and Alice did, first of all, we wanted to know, do we see changes, temporal changes? Do we see Daphnia adapting towards particular cyanobacterial strains over time? So what we did is we took Daphnia genotypes from a particular season and cyanobacterial strains from a particular season, and we did different exposures. So this is Daphnia cyanobacteria sympatric association or contemporal association versus allotemporal associations. So we crossed Daphnia and cyanobacteria from the same time point with Daphnia and cyanobacteria from a different time point. And the hypothesis is here that if you cross them, adaptation will be less. So if you have really temporal adaptation, then there's an effect of your time point in your season for adaptation. And that's also more or less what we see. So I will guide you through that. So first of all, again, important to see is here. So we have here our different cyanobacterial strains. And here we have our different Daphnia genotype strains. And you see here, survival depends on the crosses that you make. So for certain Daphnia genotypes, Daphnia will have a higher tolerance than for other cyanobacterial strains. So there's really an effect of the genotype interactions that you do, so they can deal better with particular cyanobacterial strains than with other ones. And we actually see that in the synchronous associations, survival on average is higher than in the asynchronous situation. So the A is asynchronous, S is synchronous. So in the synchronous association, you find higher tolerance. So that's also what we see here in that figure. So the other effects were not so important, but what we see is that in the synchronous combinations, Daphnia tolerance was higher than in the asynchronous combinations. So that shows that Daphnia and cyanobacteria adapt relatively fast. So within one growing season, there's even adaptation over that field season. So we repeated that for 2 years, but it was each time the temporal effect within the season that was more important. So by this we know, okay, Daphnia genotypes adapt towards particular cyanobacteria strains. So there's genotype interactions. And as I said in the beginning, we are interested in what is the microbiome doing in general. So the hypothesis that we had is that, okay, the microbiome, all microbial strains are structuring this genotype interactions of the Daphnia and the cyanobacteria. So we started to test that, and so it was Emily who started these experiments, a postdoc that came to my lab. And there, so the first thing Emily did was testing 9 different genotypes in a control diet and in a toxic diet. And there, first of all, she checked, do we see an effect of the diet on the microbial community? So do we see an effect of your diet in these different strains present? And that's actually what we saw. So that actually the phase of, it's a bit of experimental evolution on the microbiome point of view, not on the Daphnia point of view, but on the microbiome point of view. So after a long exposure to those different diets, what we actually saw is, first of all, if we compare the bacterioplankton community with the Daphnia gut community, it was quite distinct. So it shows that the Daphnia gut community is adapting slightly in comparison what is present in the environment. So we show that, okay, inoculum, bacterioplankton inoculum has an effect, but nevertheless, even there is quite a distinction between those communities. And then secondly, what we saw, that is the community of the gut microbiomes. In the gut microbiomes, we saw a relatively strong effect again of genotype on the gut microbial community. So it's a bit repeating what Martijn earlier found, but then now for 9 genotypes, so the color, different color is a different genotype, you see that there is some distinction between the different genotypes and their communities. But also, very interestingly, we also find in the bacterioplankton, so in the surrounding environment where they are occurring, in general, also a genotype effect. So the community of your bacterioplankton was also different depending on which Daphnia genotype with a particular microbiome was there. So Daphnia genotype is also structuring your bacterioplankton community. So we see that actually we have 2 levels. So genotypes are structuring gut microbial communities, gut microbiomes, which we can relatively easy understand because, okay, the microbiome and the gut is interacting quite strongly, but we also see they can modify the environment. So that's a potential for the ecosystem role we think they have. Okay, we did some additional network analysis to really understand what is influencing what, because of course it's quite complex and all these interactions are dependent on each other. So Francois helped us a little bit more there in the analysis and there. So if you look to the analysis, especially in the subnetwork analysis, we see that in here we got actually evidence via this extra network analysis that Daphnia genotype is really structuring also your bacterioplankton community. It's a subtle effect, but it's important. It's important you understand it because it's really crucial for these changes to happen. So the idea is that you have Daphnia, you have their microbiomes. The microbiomes adapt to the stressors. If they have good microbiomes and you bring them back in your environment, also depending on the Daphnia genotype that you use, you can really change the microbial community in your environment. That's actually the idea that is behind it, and that's crucial in this eco-evo interactions. Okay. So as I showed already, microbiomes, inocula affecting the outcome of the gut microbiome. There's an interaction of the host genotype with your wider bacterioplankton environment. But now, thirdly, is the question, can we modify the phenotype? Because I stressed that already in the beginning, but the question is, can we really do that? And therefore, Emily did additional experiments. So we took from this set of 9 genotypes, we took 2 tolerant genotypes and 2 susceptible genotypes towards the toxic cyanobacteria. And first of all, we compared again the microbial communities. So the idea is there, microbial communities differ between your tolerant and your susceptible genotype. And then secondly, can we modify this microbial community? So when we expose them to the toxic stressor, can we actually select for those tolerant bacteria, and then can we integrate them in Daphnia and modify the phenotype? So Emily did a big experiment on that, so I will guide you through it. It's a lot of text, but it's quite simple. So the first phase is actually, we call it's a donor phase. So it's a donor experimental evolution phase. So it's actually the same as you did with the 9 genotypes before. So we exposed here different genotypes, so susceptible ones, tolerant ones, to a control diet versus a toxic diet. So you expose your 2 different types of genotypes towards different diets. What we then did after quite some months, taking the microbial community out of the gut and then do the transplant. So we took, again, the same genotypes, but we made them axenic, so we made them sterile, and then we transplanted the microbial inocula from the donors in those recipients, and then the question is, can we modify the phenotype? So can we make a susceptible genotype tolerant? That's the question. Because if so, then we can actually save those Daphnia populations upon toxic cyanobacterial presence, that they recover from that and that they can actually keep the system healthy. That's the idea behind it. And yeah, the answer is yes. So what we saw is that, first of all, we see that the donor genotype has an effect. So how well the Daphnia was doing in the recipient. So the survival of the recipients was dependent on the microbiome of the donor genotype. So if they got the microbiome from a tolerant genotype, the recipients will do better than if they got the microbiome from a susceptible genotype. So that's the first thing. And then secondly, it also mattered which diet the donors had. So if the donors had a cyanobacterial diet versus a control diet, the recipients will do better. So if the recipients got the microbiomes from donors that had the toxic cyanobacterial treatment, the recipients will do better than if the donor microbiomes haven't seen that. So that actually also shows that we can really modify the phenotype, and it depends on which microbial community you take and from which genotype that is coming. Voilà. So that was the recipients in the beginning. Then we did a bit of follow-up to see, okay, you inoculate them and then you test them, but what is happening? So is it an initial effect, or is the effect changing over time? And that's actually crucial, because if it doesn't change over time, it doesn't, well, there's not so much interaction. And so there, Emily did some additional experiments, and what we actually saw that in the beginning, so yeah, that's a bit complex figure, but it's just important to see that in the beginning, if you give the bacterial inoculum to the Daphnia, it's mainly an inoculum effect. So what you get in your community is mostly reflecting on what you give, but if you let them interact, then actually both will matter. So your genotype effects become stronger if you let your Daphnia interact with your microbial community. So that actually shows that your Daphnia is really structuring what is being there. So and we don't know what it is. Is it a particular microbial peptide expression, immunity expression? Is it something else? But definitely it seems to structure it. It's not a fixed effect from the beginning. Okay, so that were some results we had with the cyanobacteria. Then Manon did some follow-up work, and here the stressor was hypoxia. So we brought our Daphnia in hypoxic treatment versus a normal treatment, and then Manon did more or less the same. So doing these transplant experiments, taking Daphnia from hypoxia treatment and microbiomes, putting them in recipients and see what the effect is. And again, there we have very nice phenotypic plasticity, because you can see in Daphnia, upon hypoxia, they become completely red, and that's because they upregulate their hemoglobin. So they have more hemoglobin, so they can actually deal quite well with very hypoxic conditions, which we often have in such polluted environments. And as said, the ideal situation is bring this adapted Daphnia in those polluted environments and to make them clear again. That's actually the final aim we want to do. Okay. So now I showed already, so we have this genotype microbiome interactions. There is some temporal effects, so it's not always immediately the effect there, so you need to let them interact a little bit. So we see there's also temporal adaptation. We see that Daphnia temporally adapt towards the cyanobacteria. The question is now, can you translate this temporal adaptation to spatial variations? So we saw there were asynchronous associations versus synchronous associations, and then the synchronous association was doing better than the asynchronous one. So the question now is, and that was based on a whole large-scale field study in Flanders, where we compared different ponds, and then especially in terms of urbanization. So if you have ponds that are more closer to urbanized environments, where we typically have higher temperatures, do you see different trait adaptations than if you compare it to other ponds? And then also going back to the cyanobacteria, the question actually we had here, and that's what Shira did, is so if we transplant microbiota between different ponds, will we see a higher adaptation if you give the sympatric microbiota versus the allopathic microbiota? So we did their spatial adaptation, playing with these transplants, these microbiomes, and then exposing them again to the cyanobacteria. So what you see here is that, well, it's not clear for all lines, so, but the different colors are again different Daphnia genotypes. And what we saw is that if the Daphnia got a sympatric microbiome versus an allopathic microbiome, the survival was higher for the sympatric combinations than for the allopathic combinations in most of the genotypes, not in all, but in most of them, and especially in the genotypes. So the full lines was when the donors were pre-exposed to cyanobacteria. The dotted lines is when they were not pre-exposed to cyanobacteria. So you see when the donors were coming from the same environment and have been exposed to cyanobacteria, Daphnia will do better if you give those microbiomes than others. But then, very interestingly, something we haven't expected in the beginning, but I'm very enthusiastic about that. What we also saw is that in here, if you compare the microbial communities over your different Daphnia genotypes in the sympatric associations versus the allopathic associations, you see that in the sympatric associations, first of all, the communities are more diverse, they pick up more strains, and they were more alike. So they picked up more equal strains, whereas in the allopathic associations, there was a lot of variation. So it looks like there's a bit of mismatch here between the host genotype and the microbial community. And that's according to the idea that the microbiome may help for Daphnia really to locally adapt to the different stressors here, and that there's a kind of association between your host genotype and your microbial community. Good. Yeah, I just integrated this slide because I also wanted to show some parasites. I haven't talked a lot about parasites, but of course we know that, okay, we have our microbial community here in our Daphnia guts, but we also know there are a lot of different microbiota interactions, especially parasites, as we heard yesterday also from Marcin. So in here, that's another orthospora, actually. It's also an orthospora, but it goes to the top of the Daphnia gut. We have other ones at the bottom of the Daphnia gut. These are microsporidians. We typically also have other bacteria that are in the hemolymph everywhere. That's Pasteurella, and then other microsporidians that go to the carapace. And then, yeah, there's one experiment that Amruta did in the lab, and she's coming from Justina Wolinska's lab in Berlin. So she came to my lab, and there we did some exposures of Daphnia microbiomes with different parasite treatments, parasite and control, and then also like in a time-dependent manner. I must say for the parasites, we don't find such a strong effect as we found it for the cyanobacteria so far. If we see effects, it's mostly on the total microbial community, not on the gut community, but on the total Daphnia microbial community, and there seems to be also a strong time dependence effect. So in a sense, that's congruent with what we found before. But it looks as the Daphnia parasite interactions are a bit more complex than the Daphnia microbiome parasite interactions are more complex than the Daphnia microbiome cyanobacteria interactions, which is not so strange because, as I said, cyanobacteria is part of that microbiome. Parasites, in fact, they also are, but it's a bit more complicated, I think. More factors that are playing. But yeah, staying with the parasites, so one very annoying parasite we had in the lab was an oomycete infection. It took us 10 years to figure out it's an oomycete infection, but it's typically a very opportunistic. I will wait a little. Small break. There will be a lot of lunch, I think. So this is an oomycete infection, and it kills off, like, it came into the lab, a very opportunistic one. It killed almost all our Daphnia genotypes, except for a few. And then we did experiments with those that survived. That's typically what you do. It's like taking best benefits. So then we used those 3 clones that survived that oomycete infection. And yeah, very interestingly, what we saw before, and it's also, I think, Megan's lab have been seeing before, is that if you combine stressors, sometimes they have antagonistic effect in the sense that the individual stressor is less virulent than if you have them together. So what we saw is that cyanobacteria can actually reduce the effect of the oomycete infection. But what we then also saw is that that's microbiome dependent, because we did exposures where we used microbiomes from the lab and then microbiomes from the field. And actually the effect, the antagonistic effect of the cyanobacteria and the oomycete, we only saw in this situation. So we only saw an antagonistic effect if we used lab microbiomes than if we use wild microbiomes. And we are starting, well, we try to figure out how that comes. We don't know, maybe it's because the community here was less diverse than here, and then you have more possibilities to see this antagonistic effect. But anyhow, it clearly showed that what particular microbiome you have affects other antagonistic interactions. That's actually the main thing. Good. That's the last thing, and then I will finish. So, and then to stay a bit in the theme of before from Chris. So, as I said, we want to use Daphne as like this ecosystem engineer of your environment, and we see it as a small living bioincubator that is in the pond. And then the question is, of course, we know, especially in these urbanized environments, there's a lot of microplastic waste present. So can we also use this Daphnia microbiome maybe to learn something about microplastic degradation? So the idea is that in the urbanized environments, actually you have a lot of microplastics. So what we did is we took environments with high microplastics, low microplastics, and then actually the idea is to see if in high microplastic environments your Daphnia microbiome plays a role in degrading this plastic. So if you expose Daphnia to microplastics, immediately your gut is full. So they filter it immediately. So there's a high window of interaction between the microbiome and the plastics present in the gut. And then secondly, is to see, do they really do something? Do they degrade the microplastics? Do we find microplastic degradation pathways? If we do, to look to gene functions. Yeah, that's a bit more in detail, how a Daphnia full of plastics look like. So what we did is comparing environments, high microplastics, low microplastics, and did metagenomic shotgun sequencing to see if we find different pathways. And then secondly, we also again brought them in the field, took inocula from high microplastic environments, low microplastic environments, exposed them to microplastics, yes or no, and then see if microbial communities change and if we find particular functions. It's a lot of results together. It's from a poster Anna and Broos made. It's mainly Anna and Broos who did all these experiments. But what was very interesting is, first of all, that we saw in situ, so when we looked in the field, if you have urban ponds, they clearly have much more microplastic than more natural ponds. So natural ponds were more related to nature reserve, a bit out of the cities. So definitely there has been a different level of exposure in your urbanized settings versus in non-urbanized settings. And then what we also saw, and it was especially in the urbanized setting, that we found effectively in our metagenomics shotgun data pathways for microplastic degradation. So microplastic degradation and also antibiotic resistance. So that was the two that came out most. And then also in our experiments, we see that if you expose them to the microplastics, your microbial communities are shifting. We don't have evidence yet for gene functions in the lab experiments for microplastic degradation, but that's, we think, because we didn't do our experiments long enough. So we will now do them longer. Voila, so I'm to the conclusion. So I hope I could convince you that both the wider bacterioplankton and the host genotype can shape Daphnia gut microbiomes, and that this drives adaptation of the microbiome and acclimatization to multiple stressors, and that there's definitely a scope for the microbiome to shape the eco-evo dynamics affecting host community and ecosystem dynamics. Voila, this is our facility where we have ponds and also different culture rooms. And that's then a bit non-Daphnia work, but I won't say much about that. So, but we're also now looking to snail parasite microbiome interactions. Karine from Madagascar is looking to lemur microbiomes. Thomas in drinking water distribution system microbiomes, and then Karen spider mites, and then we now started a new project also on human microbiomes. So the microbiome is a bit getting a lot. Also important for people that want to come to Europe, a very nice opportunity is, so I'm part of the ESEP steering committee, so European Society of Evolutionary Biology. We have very nice conferences in August, so very welcome to come. And voila, that is the team. And thank you very much for your attention and for following. Questions? I hope you're not too hungry.
Thank you very much. This was a really, really interesting talk for me. You alluded a few times to what specifically in these many different genotypes that you studied may have caused the microbiomes to be different. And I'm curious to see if you can elaborate on that.
Why the different genotypes differ in the
yeah, so I don't know anything about this, so maybe that's not a good question because everybody else knows. But I'm curious to know, what is it with those different genotypes that causes the microbiomes to be so different?
Yeah, no, that's a very good question. I think, first of all, well, what we see is there's variation in filtration capacity between your different genotypes. So filtration will definitely affect what you have in your different communities. So that's more mechanistically point of view. But then what we think really in this small window of the gut, so what is there in terms of receptors and expressions will determine what you finally will get in your gut. So there must be host factor related expressions that structure it. We were thinking about antimicrobial peptides. We are not sure. I also don't know if they are expressed in the gut, or that is more hemolymph. But yeah, we are doing some following up metagenomics, metabolomics work now to really see what is getting expressed there in the gut that determines these really differences. So yeah, we think it's more like associations, receptor associations for particular bacteria, or then also like secondly, and that's something else, and that's then related to the eco-evolutionary dynamics. We also think an important factor is a priority effect. So if you have a bacterial strain coming in, it attaches to particular receptors, you get a particular community in your gut, it will determine what other bacterial strains will come in. And that's then, this priority effect. So particular strains that are established there may change your final community. And that's then a more eco-evo interactions within this gut microbiome. To be honest, we don't know it yet, clearly, but that's what we think. This priority effects may, and it can explain the genotypic effect very strongly because what we think is at the step of the sterilization, we have differences between our different genotypes, and especially because we know developmental phase when you do the sterilization is very important. So if you have different developmental rates between your different genotypes, and what's very important is that we sterilize them at the moment, certain external membranes are there. So if you have external membranes and they captured particular bacterial strains before your sterilization, it will of course affect what you finally will have. So that's then a limitation of the sterilization, of course. That's why I also didn't say it's fully actinic, because we know there may be differences between them. So that could be one of the very effects we see.
Yes, very interested in your the potential for degrading microplastics. Are you continuing looking at particular species of the microbiome that are more effective at this? And also, are there size effects of the microplastics that they're would be more amenable to interacting with the microbiota?
Yeah. We are now doing longer term experiments, and what we also see, so we use different, I didn't say that, but we use different types of plastics. Also, depending on the type of plastic that you use, you get different outcome in the microbial community. So it seems to differ. Of course, also these microplastics have different capacities where they are in your system. But even within Daphnia, we see in the Daphnia gut that depending on which plastic you use, you will get a different community. And then ideally, well, what we are now doing is follow up longer term. Then we hope we can also see the pathways in the metagenomic results if we do them longer term, and then really try to disentangle. But at the moment, we are limited on what is known of microplastic degradation pathways, because each time we need to link our shotgun data to what is known already from particular pathways. So yeah, if someone can help us how to identify new pathways, I'm very open to hear that, because that would be very nice, of course. Yeah.
Thank you very much.
Yeah.
What is the source of the 9 different genotypic strains used in the transplantation experiment?
Those strains were coming from one pond, actually, so they were all hatched from resting eggs. So I didn't say that, but the very nice feature of Daphnia is they have sexual reproduction and they make the resting eggs. And if you hatch them, you're sure it's a different clone because it's a new genetic combination. So those 9 different genotypes were coming from different resting stages of one particular pond. So we took resting stages and we hatched them, and that's a variation. But so that was within one pond, but of course, yeah, over ponds, you can take resting eggs of different ponds, and then you even see more variation. And that's also what we see if we do this spatial associations, is that the further your Daphnia and your strains, cyanobacterial strains, are isolated, yeah, the better, the less good the congruence will be. Yeah.
So I have 2 questions. One is a short question, which is, on average, what's the richness that we see, or how many species do you see in the gut of Daphnia?
Yeah, good question. Let's say in terms of orders, it's like 30 orders or something. Yeah, and you have...
Yeah, okay.
Yeah, we are now writing, like, is there a core Daphnia, yes or no? Well, most people will say there isn't, but you do see, it depends, of course, if you define what is core, but in particular, especially lab conditions, we also, well, like this limnohabitant strain, for instance, one that always pops up. So there are a few, let's say like 5 or 6 strains that seem to pop up consistently between different studies, but then of course you have a lot of variation depending on what you give. Yeah.
I was just wondering, just because, in the human system, we know there are priority effects that are selected for, like by breast milk and so forth, but and my second question is, in your natural experiment with the the low and high microplastic ponds, did you see difference in the abundance of the Daphne in those 2 ponds? So when there were higher microplastics, were there lower? How does that work out?
In general, there was not so much difference, but that's a very good question. We should look at our densities again. Well, anyhow, the measurements were always done on the same number of individuals, so we standardized.
So that's different than knowing, because I thought that would be a really important thing to know, because I do wonder, microplastics can't be a very good dietary source. I assume, were they smaller when they were fed on microplastics in the lab? Did you see differences in phenotype?
No, but that is completely true, because so for pets, we expect that they will probably starve. You see it as full, but they didn't. They did relatively well. So dependent on the plastic. So for pet, they were doing quite well. For PLA, they were doing less. Did we see body size effects? I think we did, but I can't say by heart in which type of plastic. So it depended on the plastic.
And then how about the fitness in terms of the number of eggs?
Fecundity, there's definitely an effect, but in general, again, in PET, they did relatively good. In PLA, which is then supposed to be a biosynthetic a microplastic, a better one, actually they didn't do so good, so the fitness was much less. So it was a bit contrary to what we expected in terms of Daphnia fitness, yeah.
About whether we want Daphnias in natural, I mean, they're going to be there in natural ponds anyway, but, you know, in terms of what the impacts are. And, but of course, they're being eaten when they have microplastics in them as well, so.
Yeah, that's how it accumulates.
Right. And we know.
Yeah.
Really cool talk. I was interested about the temporal dynamics and rapid evolution.
Yeah.
I was wondering if you've looked at how different nutrient regimes or fluctuating environmental conditions are playing into sort of the abiotic factors into these dynamics.
Yeah, you mean if we have played with nutrients?
Yeah, like nitrogen or phosphorus or some of these elements, are they also interacting with like how these ecoevolutionary processes play out, or if you've looked at any of that?
Yeah, definitely they do. We did that in earlier experiments, but then towards the Daphnia parasites, we manipulated nitrogen-phosphorus ratios. The only thing is, it's so hard to do that. I don't know if you work with, yeah, okay. So you manipulate your ratios in your algae, and then you have to expose your Daphnia towards your different stressors and your microbiota. It's so many levels to control. It's really, really hard. But we did some work with that, and especially then related to the Daphnia parasite interactions. The result is actually dependent very much on which parasite you were looking at. With the cyanobacteria, we didn't do it yet. But that's maybe something we could look at, manipulating ratios and then see the cyanobacteria, if toxicity changes. And yeah, very nice idea. Yeah, we didn't do it yet. So, but yeah.
Yeah, really intriguing talk. I've learned a lot about Daphnia from yesterday and today, so really intrigued. And I'm most intrigued about this idea of it being an ecosystem engineer. That's really, really interesting to me. And so I have kind of two questions related to that. The first is you showed, heterogeneity between different types of Daphnia. So in the environment, in a particular pond, do we know how one might outcompete the other, and can we control that? And then the other part was you showed some Daphnia strains contain tolerant microbes to microcysteines or microplastics. And this is more open question. Have you thought about engineering some of the bugs in those guts? Because now you have the ecosystem engineer, you could maybe engineer some of the plastic degrading microbes or something, or microcysteine degrading microbes in those guts to be even better, and then really manipulated as an ecosystem.
Yeah. We are busy with that. So Emma is now doing that, especially for the cyanobacteria. Microplastics we still have to sort. But as you also indicate, it's more complex, of course, because you need to have the perfect match between the host genotype and the microbiome. So ideally, you have a whole set of genotypes, a whole set of strains, and you make your ideal combinations. But then, of course, you also need to know what strains are present in your environment to be able to inoculate that. You could also work with pools. That's what we do. So like it's a bit like antibiotic resistance, right? You try to work with pools of antibiotics, and then just to make sure that you have the one you want in it is in it. So that's one of the ideas we are playing with. And then the second problem is the upscaling. So now this is all done on small 2-liter tanks, but then the next phase, and that's what Emma is doing now, is really going upscaling and also see if that effect is there. So at the moment we are doing it in mesocosm tanks on 180 milliliters, and we are also using the ponds from the campus. So we are also inoculating it in the campus to see if we can upscale. But that's the idea. But of course, if it would work, it's a super technique. But what we also say is it won't be the only technique that is working. So first of all, nutrients have to go down. If your nutrients don't go down, it's really difficult to manipulate it because you always get them in the system. And then we're also thinking about additional techniques like algal flocculation, so combining maybe the microbiota with algal flocculation, take the algae away. So I think a best way, a combination of techniques may work. But it's difficult to say in advance if it's going to work, yes or no, I think.
Hey, thank you so much for a very inspiring talk. And I was also very intrigued by the idea of Daphnia engineering the ecosystem or the microbiome in plankton. And from a fellow Daphnia scientist, as you know, we do a lot of Daphnia parasite experiments. And I'll do a little bit of introduction. So for anybody who doesn't or haven't really seen Daphnia cultures, what we usually have is like some vessels, like beakers or jars with Daphnia inside. We feed them unicellular algae, and of course they filter it out and they change it into clumps of algae that rest on the bottom of your vessels, and then you have to clean it up every once in a while. So going to my question, what we've been noticing is that in the case of Daphnia who are infected with some parasites, we often see that those residues, those fecal pellets, look differently. So the color of the algae sitting on the bottom of the beaker tends to be, for instance, more yellow, more attached to the bottom, less floaty, let's say.
Yeah.
So what I'm wondering is, have you seen anything similar with the cyanobacteria, and do you think that's one of the symptoms of this ecosystem engineering? And how do you think it scales up? Sorry for multiple questions.
No, it's a great idea. I haven't, I must say, I haven't looked at the color, so I don't know. But it perfectly matches with what we see, right? We see that it affects the bacterial plankton community, so I wouldn't be surprised. So yeah, that's the next thing we have to do, catch Daphnia fishes and see what's in it, yeah. No, that would be really good to look more in detail what exactly is happening there. And yeah, it's what we think, because they flush it all the time, right? So you have really this, and then it's also important because you have to have this cycling, otherwise it cannot change the system. Yeah.
Hi, thank you for your talk. I really enjoyed it, and I think it's just a follow-up question to the above two questions. We see the ability of Daphnia to structure the environmental bacterial plankton community, and I wonder, what is the mechanism? Is it just pure stochastic process of them filtering and then pooping out bacteria that shapes the community, or there are other genetic or biotic interaction mechanisms in here?
Can change it. I think it's a combination of the two. Yeah, well, if you remember one of the first slides with the yellow bars, so there you see clearly you can have really strong community shifts. So if this yellow strain Citrobacter come into your environment, and that's a really a strain shift, a community shift. I think that's one of the mechanisms that happens, so really strain shift. But secondly, like I said, the phenotypic modification. So if you have a Daphnia that is more tolerant and can filter much more, it will also affect your community. It will filter more abundant ones and then maybe leave rare strains behind. So, but that's again on the community level. Can it also bring metabolites into the environment? We don't know. But yeah, ideally, if you have, like for one strains in cyanobacteria, it's known cyanobacteria interactions. You have particular bacteria that express enzymes that cleave the toxins of the cyanobacteria. So that's one of the hypotheses we have. So maybe in the cyanobacterial exposed treatments, this strain is getting more dominant, so you get a strain shift, and because of that, you get more the enzymes in your environment that cleave your toxin. And then the cleaved toxins can be additional food source for Daphnia. So that would be more enzymatic expression. I think it's a combination of the two. Strain shifts and particular expressions that help. If you have that combination, I think it's the best combination to really let the system work. Yeah, good question. Thank you. Thank you. Thank you.